So good evening, everyone. We'll get started in a few minutes. We'll give everybody about 1 more minute to get on and then we'll get started. So good evening, everyone. Welcome to tonight's seminar sponsored by the Lane County Master Gardeners. My name is Jan and I will be your host for tonight's seminar. Tonight's seminar is on management of Botrytis and all of a sudden I can't pronounce anything. Phytophthora disease of ornamental and cut flowers. Our speaker tonight is Dr. Gary Chastagner. Before we get started, a couple of items. Closed captioning is enabled and can be turned on or off with the CC button at the bottom of your screen. Also, during this session, everyone will be muted. There will be time for questions at the end of the talk. To ask questions, please type them into the Q&A box, and we will answer as many questions as we can during the time allotted. If you have additional questions, please contact your local extension office. When you leave this seminar, you will be asked to fill out a short evaluation. Filling out the evaluation will give us valuable information and help us improve what we are doing. So let's get started by introducing tonight's speaker. Dr. Gary Chastagner is a professor of plant pathology at the Washington State University Research and Extension Center, New Puyallup, Washington, where he has worked since 1978. He received his Ph.D. in plant pathology from the University of California at Davis in 1976. He leads an internationally recognized research and extension program dealing with the management of diseases on ornamental bulb-cupped flower crops and Christmas trees, management of sudden oak death, and factors that affect the post-harvest quality and safety of cut Christmas trees. I'll turn it over to Dr. Chastagner.
Thank you very much. It's a pleasure to be here, and we'll share some information that we've gathered from some of our research. So this presentation may be a little more researchy than some presentations that you listen to, but I'd be happy to try and answer questions at the end. In addition to myself, any business, any group, you're only as successful as the people you have working for you and stuff. And I'm very fortunate that over the years I've had a very good staff. Two really important people right now are Mary Ann Elliott, who's a research associate, been working for me for a little over 10 years, and then Joey Hulbert, who's a postdoctoral fellow, has been working in the program for the last couple of years. So WSU, the main campus is located over in Pullman, which is on the eastern side of the state. I work at the research at a research and extension center in Puyallup. We're about 35 miles south of Seattle. And when I was hired to work on ornamental bulb crops and turf grass. That's changed over the years. I still work on ornamental bulb crops and cut flowers, but we do a lot of work on Christmas trees and increasing amount of work on various types of trees in urban landscapes, as well as indicated, working on sudden oak death. We've worked on that for almost 20 years now. Most all of you are aware that, disease management is really based on principles and things you learn about the disease triangle or disease pyramid. You have to have a pathogen present, you have to have a host, and a susceptible environment for disease to occur. The other aspect of it is you have to have time, because if it takes, let's say 12 hours, 24 hours for infection to take place, and then it might take another few days to a much longer period. Some of the conifer diseases we work with, it takes almost a year for symptoms to develop. So there is the time aspect of it as well. So a lot of questions come up as I've got plants that have some symptoms. They might be being killed. So what is killing my trees, or what's killing my plants? Why is it wilted, etc. I think everyone realizes that there are abiotic problems. They're not caused by a pathogen. In some cases, these are small root systems from Christmas trees planted in a field. And you can see there were girdling roots here that were basically cutting off the roots and causing the trees to wilt, much like they would if they had a root rot. Or you could actually have an actual biotic disease caused by generally a fungal or fungus-like pathogen, such as our malaria root rot or a Gnosis root rot or Phytophthora root stem canker disease. So we use a variety of approaches to identify what is the cause of a problem. We are not always successful in doing this, but generally there's traditional approaches where we would examine samples with microscopes, a culture from symptomatic tissue, and then look for spores or the colony morphology. More commonly now, we're also using a lot of molecular diagnostics, sequencing of DNA to identify pathogens. And in some cases, you can identify things from cultures or symptoms plus the signs might be fruiting bodies on the plant. But in some cases, you have to basically submit a sample to a plant clinic or lab that can do the laboratory diagnosis. As I indicated, I've worked on ornamental cut flowers, bulb crops specifically, for almost 45 years now. This is one of the legacy programs here at this research center. And there's an increasing number of small farms producing and selling a wide variety of cut flowers. Here's just some pictures from the Seattle Pike's Place market and the Seattle wholesale market. In addition, these cut flower farms also, in some cases, add significantly to agrotourism. Picture on the left of the Skagit Valley Tulip Festival, the Wooden Shoe Tulip Festival in Woodburn, and Holland American Flower Gardens in Woodland, Washington. A number of years ago, we did an economic assessment of what the value of the cut flowers were in Skagit Valley and compared that to the value of agrotourism. At the time of the assessment, the value of the cut flowers was about $25 million annually. The agrotourism associated with the Tulip Festival was around $84 million annually. So there was actually more value to the community from the agrotourism component than from the sales of the crop. So one of the things you realize when you start working on cut flowers and particularly bulb crops is that they present a great challenge as far as integrated disease management because they are so diverse in the crop problems that occur on them, and often the entire plant must be aesthetically pleasing, whether this is being used as a cut flower or in a landscape situation or in a park type of a situation. There's virus diseases, fungal diseases that are foliar in nature, fungal diseases that cause decays of the bulb in storage. And so there's just a tremendous variation. There's some of the soil-borne diseases that once you have the disease in a field, these also are important in landscape situations at parks, for example, where tulips are planted year after year in the same site, that the inoculum builds up and it can persist for a number of years, and so it makes it very difficult to successfully grow another crop of tulips there. Let's see. One of the pathogens, Botrytis, and there's a variety of species on bulb crops, and these tend to be highly pathogenic, and they are probably the most common fungal disease on many of the bulb crops that are produced worldwide. Lilies, tulips, daffodils, peonies, iris, a number of these crops are impacted severely by the death of the foliage, and they can affect the post-harvest quality of the flowers and how long the flowers last and the marketability of those flowers. This is a busy slide. I apologize for that, but I wanted to show you some information. So Botrytis was first named as a genus in 1729, so a long time ago. And species were described based on morphological characteristics, such as the spores that were produced, the cultures, how the cultures looked in isolation in the lab, what hosts they were on. And so up until about 1973, based on these morphological characteristics, there were 380 species of Botrytis that have been reported in the literature. That's a lot of species of Botrytis. Part of the issue is when you look at Botrytis, there's a lot of variation in the growth of Botrytis. So these are 3 isolates of new species of Botrytis that we found on and named. It occurs on peonies as well as grapes. It's been reported in Italy as well as our detections up in Alaska. And so each of these is a different isolate of Botrytis euroamericana. So they look different. They look different in culture. Only one isolate out of the 3 produced sclerotia. So in the past, this variation would lead to someone naming one isolate something and another isolate another species. Same thing here. Botrytis prenorum, which is a fairly recent Botrytis species, and Botrytis cinerea, which is probably the most commonly known species, commonly referred to as the pathogen that causes gray mold on a wide range of crops. But if you grow these isolates, the same isolate here on different media, you can see that it behaves very differently and stuff. And so this also led to proliferation in the number of species that were described. But since the 1970s, there's been a lot more molecular work done using DNA and using molecular markers to identify species. And so now, so when all of this work was done with these previously described species and looked at from a molecular standpoint, there were about 30 isolates of Botrytis species, or 30 species of Botrytis that were identified. That has stayed fairly constant, except we have new species that are being identified each year. And so the increase in the number of species is largely because of the use of these molecular approaches. And today there's probably about 40 species that have been identified, and a number of novel species that have potentially been identified but just haven't been named yet. So if we look at Botrytis life cycle, or the disease cycle, it survives the sclerotia over the winter. These are hard structures that allow it to overwinter. These sclerotia during the winter, they are fertilized, and then in the spring they'll germinate if they're on the surface, and they'll produce conidia. And these conidia will blow to nearby surfaces of plants where they may cause infections. And then you get those infections during the growing season, and they'll start sporulating, and spores will be produced. And so you have this cycle that's occurring during the summer or the growing season, where it's just repeating production of spores, infection, more spores, infection, and so the disease can build up fairly rapidly. Some of the diseases that Botrytis species cause on bulb crops are referred to commonly as fire, is the common name of the disease, as opposed to gray mold, and that's really a reflection of the speed at which the disease can build up and the destruction and how fast it can occur on some of these bulb crops. It's interesting, if you look at disease management practices and how they've changed over the years, fungicides play an important role in the management of fungal diseases, and particularly on bulbs and high-value cut flower crops. And the number of fungicide applications, the percentage of the crop grown that's treated with fungicide varies tremendously. This data, and I apologize, it's not very clear, shows that a high percentage of the daffodils, tulips, iris, and lilies being grown or treated, and applications typically are ranging from 1 up to more than 30, particularly if you're looking at Easter lilies in the southwest corner of Oregon, down the border of Oregon and California. Obviously, the technology of producing bulbs has changed a lot, as has disease management practices over the years. We've done a lot of work. One of my goals is to one of the major focuses of my program is to help growers improve their disease management practices. And part of that is reducing the frequency of applications of fungicides if they're needed, to just when they are needed. And we've been able to do this on Botrytis on tulips and Botrytis on lilies in commercial fields. And we're basing sprays on the crop development and not the calendar. So the crop development, and by basing the sprays on the crop development, we've been able to reduce the frequency of applications by about 60% in both of these crops. Recently, we've started working, I say recent, it's 2013, we started working on peonies. I hadn't done a lot of work on peonies. I was invited to participate in a project up in Alaska because there was increasing production of peonies as a cut flower in Alaska. So we started doing work on peonies. There's a diversity of Botrytis species that occur on peonies. They cause foliar diseases, stem cankers, kill the stems as they emerge out of the ground, and cause post-harvest losses as well. I was fortunate enough to have a graduate student, Andrea Garfinkel, who was working on a Ph.D. in my program, and she worked on Botrytis on peonies. We identified a new species, Botrytis euroamericana, and then she also identified 10 previously undescribed species that primarily are present in Alaska, and we don't see them when we've done sampling in the Netherlands, in Oregon, or Washington. So there's something about the Alaska boreal climate. It's good for growing peonies, but it's also has a tremendous diversity of Botrytis species, and the peonies are almost behaving like a trap crop up there. So one of the things we've looked at is susceptibility of peony cultivars to Botrytis. We've used 3 species of Botrytis. Botrytis peony. Generally, when you see a Botrytis with a name such as peony, that means it's a host-specific Botrytis that generally only occurs on that host. So in this case, peony. Tradus Tulip, as you might guess, is a host-specific species that occurs on tulips. The Tradus cinerea, as I indicated before, is a very cosmopolitan species that occurs on many, many different hosts and it's very, very common and commonly associated with gray mold. And then we've looked at some new species that haven't been described yet. We do inoculations on cultivars and foliage of cultivars, and then rate for disease development. And there's tremendous variation in the ability of the Botrytis species to cause disease. Unfortunately, we have not seen a lot of variation in the cultivars that we've looked at. So under favorable conditions, conidia that are produced on these disease leaves can rapidly spread the disease in the field. It can, the initial infections are observed as the shoots are coming out of the ground. And if you have cool, moist conditions, or if you have a hailstorm that damages the foliage during the growing season, you have significant increased potential for disease development. So if you were to look in the Pacific Northwest Disease Management Handbook, you would see a variety of cultural controls that are listed. These are very important standard practices that are used in managing diseases. Unfortunately, we don't see a lot in the way of what I indicated, resistance in cultivars and stuff. And the reality is sometimes horticulturally desirable cultivars also can be quite susceptible to some of these diseases. So growers face challenges. If you have a cultivar in a garden situation that happens to be very prone to being susceptible, you may face challenges in a garden. Sanitation is extremely important with peonies. The petals from the flowers, if you had flowers, plants in a landscape situations, allowed them to flower, you weren't harvesting the flowers or left some on to open up. Once those petals start falling off those flowers and land on leaves, Botrytis develops very rapidly on those petals, and then it spreads to the foliage. So generally, sanitation, removal of flowers off of plants prior to the loss of or dropping of the petals is an extremely important aspect of managing botrytis on peonies. As I indicated, chemicals are commonly used in some production systems. In Alaska, there were a lot of growers that were putting in farms or plantings, and they preferred not to use chemicals, or they preferred to use organic-based materials, and I'll show you some information about how effective some of those types of products are. I also indicated that post-harvest disease management, disease can develop during storage, and so post-harvest disease management is really dependent upon your disease management prior to harvest. You need low levels of disease on any material going into a cold storage facility, reduces inoculum, reduces the potential of disease development on stems. In the storage conditions, particularly temperature and the duration, are important. Sanitation, keeping storage facilities clean is really important. And then there are some post-harvest treatments that are potentially look like they'll be effective in controlling disease. One of the things we do is I've run disease control trials on particularly tulips. For tulips, we've done, I think, almost 45 years of continuous disease control trials under field conditions. We do some under greenhouse conditions as well. These are a list of products that we've tested. All the ones with arrows are biological-based products that are being developed or are available for use. Sometimes they're recommended for use to control botrytis. The other materials are standard types of fungicides. Some are traditional type fungicides that are used, such as Spores, which is a mancozeb product. Others are new materials that are being developed through the IR4 program, and we're constantly screening materials to try and identify both biological-based and traditional fungicides that are effective in managing these diseases. So one of the problems you have when you manage diseases with fungicides is there's always a question if the fungicide application didn't work, do I have resistance to that fungicide, which for some fungicides can be a significant risk, or did I get good coverage, or was the timing wrong? Did I already have infection? So to eliminate that, one of the things we do initially in screening some fungicides is we'll do a dip in the fungicide, take the leaf, dip it in a suspension of fungicide. There's no question that there's coverage, and then we'll do an inoculation of those with the botrytis. And in this figure here, we have lesion widths associated with. Here, we have an inoculated check. It has the largest lesions, but statistically, all these treatments that have an A in front of them were no different than this inoculated check. So we
Gary,
have. Yes.
Gary, I'm sorry to interrupt. This is Sharon Roberts. We're not seeing your slides advance. The current slide is the one, disease management, with pictures of harvest treatment through the years.
Okay.
Yeah, sorry.
Let's see. How far back was that slide?
Yeah, sorry, it wasn't clear, and then you referred specifically to a figure. I'm wondering if we go into slide sorter.
Okay.
Sorry to interrupt, but I know.
No,
like
hey, if you can't see the slides,
yes.
Can you see the slides now?
Yeah, that one is the agritourism slide.
Yep. So let's see here if we can. You see that?
Yep.
You tell me when you see the slide that.
Okay, the last one that we saw. Yeah. Keep going. There, that was the last slide. Yeah, we had not, that disease management was the last slide that we saw.
Okay. So I'll just briefly. So is the next slide? Are you seeing still the disease management slide, or is there another
No,
slide
now
that's
I'm
there?
seeing phenology.
Yes. Okay. So this is where I was talking about the crop phenology, basing those sprays on tulips and lilies. We were able to reduce sprays by about 60%. This is just a series of pictures of Botrytis on peonies, foliage on the stem. It's attacking the stem as they're coming out of the ground, gets on the flower buds. You can see some infections on buds here that are quite small. If those go into a storage facility and they're held for any length of time, this is what you get. So a gray mold developing. This is a picture, Andrea Garfinkel here in the back, and Katie Coates, who is our molecular lab person, showing the Botrytis species that we were working with. These are the ones that are reported to occur on peonies, and then we have these novel species, primarily in Alaska. This was the susceptibility trial showing how we inoculate the leaves. On the left, this is Andrea here, and then Dr. Pat Holloway from the University of Alaska Fairbanks, who's a collaborator on our project. This shows the difference in the ability of this species to cause disease on peony foliage. So as you might expect, Botrytis peony is a very aggressive Botrytis species. Botrytis cinerea causes a significant amount of disease, as well as one of these new species that hasn't been named yet. Another life cycle picture just showing the importance of spores being produced. I can't tell you how many millions of spores are being produced on this blighted flower here. There are literally millions of spores being produced that are spreading with air and can rapidly spread the disease. This is a slide I showed in relation to the Pacific Northwest Disease Control Handbook. If you look in there, typically they'll have a description of symptoms. They'll have a description of cultural controls. There's a list of cultural controls. And really the management of plant disease is the foundation for the management of plant disease is the utilization of cultural controls. This is why understanding the disease triangle and the environmental factors and things like this, the pathogen life cycle are so important because you can utilize these cultural approaches. This was a slide showing the colonization of the petals that had fallen off the peony down onto the leaves. They were colonized by Botrytis, and the Botrytis spread then to the foliage. Chemical controls, most fungicides do not kill the fungus. If it's already present, they protect plant tissues from infections. There are a few systemic materials, but most fungicides are protectant in nature. And as indicated, we're doing a lot more work now with biological-based materials. It is also important whenever you listen to a talk such as mine to realize that sometimes I'm using experimental fungicides. These are not registered for use on the crop that I'm experimenting on. And so it's always important if you use a fungicide to follow the label and make sure that the crop and site you're applying the fungicide to is on the label. And then storage conditions post-harvest as well. This was the slide that I was pointing out where the arrows were pointing out the biological-based materials that we've been testing or have tested over several years on both tulips, lilies, and in some cases peonies, as well as the traditional fungicides. I'm always looking for the FRAC groups on fungicides. This tells you what their mode of action is, and so a material, even though it may be a FRAC group 11, you may get another material. It has a different sold as a different name, but it actually has the same biological, the basis of the activity is the same, and so resistance can be an issue if you're using materials that have the same frac codes. This is the slide where we were at when you said that you weren't seeing slides. So on the far left is the inoculated check. We analyze this data statistically, and so any of these columns that have the letter A are no different than the inoculated check that wasn't sprayed. So you can see all of these treatments were not different from the inoculated check. Conversely, these materials out here, which do not have the letter A, all of these are significantly less smaller size lesions than the inoculated check, and some of these are very little, if any, lesion development. The arrows here are pointing out all of the organic-based materials. And this was when the leaves were dipped in these materials, so there was no question of coverage. So in general, the organic materials that were included in this trial did not do a very good job of reducing lesion development. We've also looked at a number of other materials here. This just shows a list of the materials, and as indicated, the frac codes. It's important if you're recommending or using fungicides to pay attention to the frac codes. The same thing is true with insecticides. So this is disease management during the growing season, and we go out, we apply materials here. So here the fungicides were applied, and then we rated disease development, and then we calculate the rate at which disease development, it's a term called area under disease progress curve. So we have the inoculated check. So it had the highest area under the disease curve. That means that the disease developed first on those plants and developed more rapidly on those plants. As we go down here, we have some treatments where very little disease developed. Again, the A's here are not different from each other. These are not different from the non-inoculated check, where we had very low disease, and the other materials. So post-harvest problem with Botrytis on peonies is a big issue, as I indicated. Here's a bunch of peony flowers, stems that have been stored, and you can just kind of see all the decay that's occurred on those during storage. Hopefully this isn't happening if you're a commercial grower. One of the questions we had during our experiment is, what is the source of infection during storage? Is it infected buds that the grower's not able to sort out because they don't see them? Obviously, these infections are large enough that growers would see them. But in some cases, you might have a single small infection that the growers don't see. Or is it airborne conidia? So what we did is there's some selective media that you can use that Botrytis only grows on. So this is a really good tool. And all the Botrytis species that we've tested will grow on this media. So we can take this and put these plates on a bench or a shelf in a cold room, take the lids off and expose them for 1 hour, or expose them overnight. And then we come back, collect them, put the lids on, incubate them, and then count the number of Botrytis colonies. So these are different cold rooms in Alaska that I sampled, and the dates at which they were sampled, and the number of Botrytis colonies that developed. So they were spores, as you would expect, in the air. So even if you took a bud into the cold room that had no disease on it, and didn't have any spores on it that you brought in from the field, there's spores in the air that will be deposited on those flowers. And if you have conditions favorable for infection during cold storage, you have disease development. This is temperature. We were doing some temperature, moisture, and modified atmosphere storage. You can see if you have high humidity, even at temperatures of about 34 degrees Fahrenheit, with prolonged storage, you can see that the Botrytis just starts at where the flower buds are and starts consuming the foliage and stuff. Here's the difference if you have similar leaves that have been, or similar stems that have been treated with fungicides prior to harvest. The reason being is that most all of the Botrytis species that you see on peonies and on other flower crops, they're able to grow at temperatures almost down to zero. This figure doesn't go to zero, but you can see they don't like real warm temperatures. Their optimum's about 20 degrees, which is 68 degrees Fahrenheit, but they're even growing down here at 5 degrees, which is about the temperature inside of a refrigerator, and they'll even grow more slowly, but they will grow at zero. So even in cold storage, you can have growth of the Botrytis, and so the longer you store the plant or the stems, the more problems you have. So one of the things that temperature fluctuation in cold storage is an important variable. And so all of these figures are basically the same. This shows the humidity or the range here in temperatures and the humidity. Here's a cooler where there's a lot of variation. This is bad. Anytime you have bad, things warm up, you get condensation, free moisture, which is necessary for infection or germination of the spores. So if you have coolers that have variation in temperature and or humidity, that's not desirable. Here's a series of coolers. You can look at some. They're very good as far as consistent temperatures. Others, they're going all over the place and they're much higher temperatures. So the higher the temperature, the more likely you are to have problems if you're trying to store the crop. So post-harvest management, it's really about understanding your environmental conditions under which you're storing them, maintaining that at optimal storage temperatures, which is as close to freezing as possible. We've done air shipment of plant stems to see what happens during shipment. And these are stems that were shipped from Homer, Alaska via air to me. And so you can see they warm up and then they get to Tennessee and the temperatures goes down. They're there at night. And on the shipment, they're to me in Puyallup, they're going up. So when you start shipping flowers, you have the risk of increasing the potential for disease problems to manifest, so that you may have a box of flowers that you loaded into let's say a FedEx shipping thing. And by the time it gets to the consumer or the florist, you've got problems. We've done a lot of storage trials as well. Temperature, you can actually freeze peony stems down to about -1 degree centigrade, 29 degrees Fahrenheit. That would improve storage and reduce disease development, but it's really touchy to be able to maintain that. We've looked at some fungicides, can screen fungicides using the flowers. Applications of the fungicide, then spray the spores on the fungicides and identify fungicides here. As you can see, some have disease, some do not have disease. This is a good way to test biologicals as well as traditional fungicides. It's a fairly rapid test that you can get data in about 24 to 48 hours. And what you see here is, here's a biological, didn't work. Another biological and stuff. It's less than the inoculated check, but it's still fairly high. Here's a number of traditional fungicides. There's a reasons why growers tend to use traditional fungicides with high value crops such as cut flowers. In general, they're much more effective, and they also provide good residual control. We do a lot of post-harvest trials. We have a large 2,000 square foot temperature controlled post-harvest display room. We just wrapped up a trial today. In fact, we'll start another one in about 3 weeks. And Botrytis here can cause lots of problems on flowers during display, as you see here. You can have ones that don't have much Botrytis, and you can have ones that have Botrytis. We rate the severity of Botrytis on the flower or what's left of the flower. And when we do this, if we have a pre-harvest spray, some materials like Austin work very well. If you do a post-harvest treatment of the flowers, you have a few more choices of materials that work very well. This is just another example. Excellent disease control on flowers during storage. Long-term storage, we're going out 12 weeks, so growers can take advantage of improved value of the flowers. Disease just continues to develop. The longer you go, the more disease pressure you have. So we've been looking at some fungicides and materials, and some of the materials look really well, give us flowers that we can still have good quality after about 8 weeks. The temperature duration is super important. We do have a new fact sheet on peonies. We actually have two, one on tobacco rattle virus and one on the most common diseases that you see on peonies. And so I'm going to make a quick shift here, and we'll go through some stuff on Phytophthora. We spent a lot of time on Botrytis. I apologize for the problems with the slides and stuff. So most of you are probably aware of Phytophthora. You're probably aware of sudden oak death, which is caused by Phytophthora amorum. It's a significant issue that nurseries and the forestry community is dealing with, particularly in California, nurseries in Oregon and Washington, and some forested areas in Southwest Oregon are also dealing with Phytophthora amorum. And Phytophthora, the name Phytophthora means plant destroyer, and it was initially described in 1875, so about 100 years after Botrytis was described, the genus was described. It's responsible, Phytophthora infestans, very common disease on tomatoes and potatoes. It was responsible for the Irish potato famine in the late 1940s, where millions of people starved to death, resulted in the large immigration of Irish people to other countries, including the United States. The unique thing about Phytophoras is they tend to produce these swimming zoospores. I'll see if this works here. Looks like it's going to. So this is a sporangium. Instead of having air-blown spores, when there's free moisture present, the cytoplasm differentiates within the sporangia into spores that have flagella or little tails on them, and allows these spores to swim. And so when there's free moisture present, the sporangia will germinate, produce zoospores. Those zoospores are attracted to chemicals being released by the growing root tips. They insist there and infect those root tips. So it's a really sort of very effective way for this pathogen to be able to get to and infect roots. Not certain how come so life cycles, there's a series of structures, spores, thick-walled, either chlamydospores or oospores if there's a sexual stage, that can survive over for extended periods of time. They can germinate to produce sporangia, which can either directly infect or can produce zoospores, and then you have infection, more sporangia produced, and a cycle that's just repeating an increased disease during the growing season. A lot of Phytophthora issues associated with international trade and the importation of the introduction of exotic pathogens like Phytophthora morum. These then get into nurseries and are disseminated with the movement of infected plant material in nurseries. So there's been a tremendous amount of work trying to improve the health of plants in nurseries to reduce the risk of Phytophthora developing in nurseries, as well as the risk of spread of Phytophthora. Sudden oak death, as I mentioned, was a new disease. In the early, 20 years ago, 25 years ago, it has killed millions of trees in the forest in the coastal areas in California. It's present it can occur on a number of ornamental plants. Rhododendrons, viburnum are just a couple of the nursery stocks that are extremely susceptible to this. When there is a detection of Phytophthora morum in a nursery or a landscape, this triggers state and federal regulatory actions and mitigation activities to limit the spread of this pathogen. So this is a picture at the Bloedel Botanical Reserve on Bainbridge Island in Western Washington. We notified APHIS and the regulatory agencies of detection of Phytophthora morum at this site on a Friday. There were survey crews on the ground on Tuesday morning, delineating where the disease was present. And then a lot of our research relates to mitigation, and we're doing a lot of research as it relates to steaming infested sites. So infested sites are identified, plants are destroyed, removed in a landscape situation or a nursery situation. If there's any chance that there's inoculum in the soil, we then typically are steaming these sites. In some cases, this means that we have a steamer, a portable steamer that we use, that we have to be able to steam a site that's 300, 400 feet from where we can get the steamer. So over the years, we've developed techniques of delivering steam to these remote sites to do this. We've also developed techniques when we have a large nursery where there's positive remorum detections, such as this one, and large destruction blocks. So thousands of plants being destroyed and soil or the gravel beds being infested below these. We have been working on developing and have developed the ability to steam and kill the inoculum of the pathogen in large areas. So there's a series of very good management practices, safe handling, integrated pest management practice 1 through OAN, and that are available. Anyone in the nursery business should be practicing these integrated pest management practices and doing everything possible to reduce the risk of Phytophoras, not just because it's a regulatory, in some cases there's regulatory concerns, but because it will help reduce losses from the Phytophoras that are not regulatory pathogens, but are fairly common in nurseries. I'm going to quickly here in about 5 minutes go through Phytophthora on Christmas trees. The Pacific Northwest is a major production area for Christmas trees in the U.S., produces about 40% of all Christmas trees. The most common species grown is noble fir, which is extremely susceptible to Phytophthora root rot. The most common species nationally that is grown is Fraser fir. It's also extremely susceptible to Phytophthora root rot. These Phytophoras can occur on nursery stock, conifer nursery stock, such as seen in this picture. And you can imagine what would happen if you tried to lift stock out of a field such as this and then transplant it into either a forest site or a Christmas tree site. You're going to be introducing you're likely going to be introducing the pathogen into these sites. Katie McKeever, who's a Ph.D. student in my program, did a lot of work screening true firs, like of alternatives to Noble and Fraser fir for resistance to Phytophthora root rot. Part of that meant we had to have an understanding of what the Phytophthora species were in Christmas tree plantings across the U.S. So she did a lot of sampling in various Christmas tree production regions and found 14 different Phytophthora species associated with root rot, and show that there were regional differences. So the Phytophthora species that were causing problems in North Carolina were different than the Phytophthora species that were causing problems in Western Washington and Western Oregon. So because these Phytophoras they're considered water molds because they produce these swimming zoospores. Poorly drained sites are very conducive to disease development. And so you have spread because of infected seedlings, contaminated soil being moved around, or it's spreading in water. There's a series of pictures I took in North Carolina. North Carolina has a lot of Christmas trees up in the Appalachian Mountains. And here's a picture from up on the hillside, Christmas tree stand. Water runs from the Christmas trees down to a collection pond, and there's some diseased trees in the Christmas tree stand. There's a conifer nursery bed right next to the disease or to the collection pond. Water's being used out of the collection pond to irrigate the nursery, and what do you know? You get Phytophthora. And so there's a vicious cycle here, or has been in the past, a vicious cycle of just bringing more disease, planting stock into sites and increasing the amount of disease. Fortunately, that is not happening as much as it used to. These Phytophoras that you see on conifers can also cause infection of the branches and the foliage, and you get these aerial cankers associated if you irrigate with infected or infested water. You can get direct infection of the needles and the branches above ground. It's not a root rot. You can screen seedlings for Phytophthora, and we've done this. This is a picture of Fraser fir. Again, cultural controls, very few chemical controls that work even in nurseries, and they do not generally work and are not recommended for using in Christmas trees. But what is recommended is site selection and planting resistant species or using resistant species as rootstocks. Unfortunately, in the past, most of the resistant species were species of other types of conifers that were not necessarily that most desirable as Christmas trees, such as spruces. So what we observed over the years is that if we look at noble fir, this is a planting I have at Puyallup, or had at Puyallup, Phytophthora was killing these trees. Here's a row of Nordman fir. They didn't seem to be affected hardly at all. So Katie took all of her isolates that she had, ran a series of tests to look at what happened as far as if you inoculated a variety of different species with a different Phytophthora species, a variety of different conifer species with the Phytophthora species that she found during her surveys. And what happens if you have soil that's a high temperature soil like you might have in North Carolina versus our cooler soils that we typically have here? This was a big project. Looked at 7 conifer species, 4 different species of Phytophthora, 3 isolates of each one of these, and then 2 greenhouse temperatures, one less than about 70 degrees and one greenhouse temperatures between 80 and 90 degrees. So a lot of work, as you can imagine. Each one of these individual plugs had to be inoculated with a specific Phytophthora species, and it had to be done twice during a 16-week period. Oh, here's symptom development in the cool greenhouse. Symptom development in the warm greenhouse. Obviously, the temperature at which the plants are grown had a big effect. You can see mortality. These are Nordman, a noble fir, this is a disease development on noble fir and Fraser fir. Nordman, Turkish fir are down here, and that's fairly consistent. We had much higher disease at warm temperatures than we did at cool temperatures. Part of that is because of increased transpiration of the host, but also you have increased growth of the pathogen. So we're just about done. What happens when you take the stuff up to the field? If you create what we call a rice paddy, so we infest soil with Phytophthora, plant seedlings, then flood irrigate it for 48 hours. No one would do this. You're creating ideal conditions, saturated soil conditions. The zoospores are swimming around causing infections. You drain the field, then you let the new growth come out, and you monitor disease development. So you can see a spattering of diseased trees here. Well, we had Fraser fir and noble fir in this trial. Here, these pictures are with Turkish fir. Here's the amount of disease on the Nordman or the noble and Fraser fir. Here's the amount of disease under the severe conditions as far as their conduciveness for a development of infection and disease on the different sources of Turkish fir. So there's some of these species that are now being grown, such as Nordman and Turkish fir, that are very, I won't say they're totally resistant or immune, but they're very tolerant to the Phytophthora species, much more so than the Fraser and noble fir. We see this, two rows here of Nordman fir and noble fir. Big difference. Much larger trial. Many of the true firs are quite susceptible to Phytophthora. Some are quite tolerant. So what we're seeing in the industry and sites where you can't grow noble and Fraser fir, because of this work, there's tremendous increase in the utilization of species such as Nordman and Turkish fir. And we're doing work right now on Trojan fir. But with that, I'd just like to say that if you were to go visit our website, you'll also see that we have a Forest Health Watch program. A lot of information is there on some of the recent work we're doing in forest, including recent work with sooty bark disease, which is a new disease that we're seeing particularly on maples in landscape situations. And a lot of this work is, of course, supported by a number of different agencies and groups. Without their support, both in kind as well as financial, none of this work would be possible. So I've run quite long. I apologize for that. We do a lot of work in Alaska, and it's nice to be up there in the wintertime and speaking to grower meetings, and sometimes we get an opportunity to go out and see a dog sled race. So I'll stop the sharing and see if we can answer any questions.
So I have a couple of questions. One person wrote in that they didn't catch the definition of FRAC code, F-R-A-C code.
That's the Fungicide Resistance Action Code. So if you type in FRAC codes for fungicides, it'll take you to a website that lists all the fungicides that are available, and it'll tell you what that FRAC code is. So you can identify which fungicides have the same mode of action. And if you were trying to manage resistance, you would not use those same materials in like a rotation program, because in essence, you're putting the same product on.
Okay. Another question, if a fungicide is a plant protectant rather than killing the spores, how does it function as a protectant?
Generally, what it does is it prevents the spores from germinating or prevents the germ tube from infecting the plant tissue. So there's two steps there. It can affect, the germination of the spore. It can affect the germ tube growth and subsequent infection of the tissue.
Okay, any other questions out there?
Yeah, I was curious, when you were talking about peonies, you mentioned that there are resistant cultivars, and I was thinking, many of our gardeners, of course, are thinking about things in their own landscape. So when we go to the, the plain old retail nursery, are we finding more and more resistant species or cultivars of peonies?
No, unfortunately, there's not very many resistant species, okay? And in fact, some of the newer species, such as the coral series, the coral sunsets, the coral charm, which are very attractive flowers and stuff, they are extremely susceptible to botrytis. The key in a landscape situation is you can do the sanitation necessary to reduce the risk that you have of botrytis development. It's very difficult if you have, a 2-acre field of peonies. The largest peony field I've ever been in is 150 acres. You're not doing sanitation in that field. You do sanitation at the end of the year when you cut the foliage and you remove that foliage or burn that foliage, but you're not doing sanitation during the growing season.
All right. I would just like to say thank you, and also mention for everyone who is attending, this seminar in July was sponsored by the plant diagnostics specialists, and I'm one of them. And I want to just say that I really appreciate the opportunity to sort of see that the kind of research that goes on, behind, in a sense, for us, behind the scenes. How do we get the information that we then see in the PNW? So
right, it's through, ultimately the research is translated down to things like the fact sheets that come out, or the plant disease management guides and stuff like that. But it's a lot of work, as you can imagine. It's a lot of fun work. I have a blast. I've been able to travel around the world and doing collaborative things and stuff. And I get to see some neat people and who are enthused about the crops they're growing and the plants they're growing. So it's a lot of fun.
Wonderful. And thank you for sharing that with us.
Yeah.
And for the fact that now we'll get Christmas trees that are in better shape.
Yeah, well, I mean, one of the things that we do, and I could do a talk sometime on how to care for a Christmas tree, because one of the biggest issues with Christmas trees is shedding of needles, and we've been able to identify individual trees and sources of trees that do not shed. Even if they're harvested, no one will be harvesting them, but sometimes we run our trials in late September to identify sources that don't shed, because needle retention on Christmas trees is dependent upon cold acclimation prior to harvest. And so if we have warm falls, that's going to increase the problems we see as far as needle retention on Christmas trees. So we're working on identifying selections of trees or individual trees that do not genetically have that are not predisposed genetically to shed.
Thank you. That was a great presentation. Thank you for so to wrap it up, just a reminder as you leave the seminar to remember to fill out the short evaluation, and please join us next month on Tuesday, August 16th at the same time for a seminar on growing fruit trees in the Willamette Valley. Thanks, everyone. Thanks.
Bye. You guys have a great day.
Thank you.
You
too. Appreciate your taking the time.
Yep.
Bye-bye. Should we turn off recording? Oh, somebody will.